Thank you, Dana, and the rest of the speakers. Dolly was incredible for us as an institute, and this is not part of my talk, by the way. What happened is we got absolutely inundated, tidal wave of journalism and journalists about it. And the best story I have in that regard was, like every organization, you have your phone number is very simple. It's the last three digits. Well, most organizations, last three digits change. And the journalists very quickly got hold of this because we were very tight on who communicated. It was our director at the time, Harry Griffin, and Ian Wilmot, who were doing the communications. Well, the journalists got hold of the fact, and they phoned in, and it went into one of the service rooms at the back of the building at one point, where the cleaners were sitting having coffee, and the lady on the phone said, "Is that the Roslin Institute?" And the lady said, "Yes." And she said, "Do you know Dolly?" I don't think so. I've only been working here a few years. And brilliant. Anyway, I'm slightly different from the previous talks, which I thought were excellent. I'm just going to tell you a little bit about what's possible in livestock. I'm going to take you through a very sort of early, very brief charge through agriculture or livestock agriculture and breeding to sort of set the pace. And then I'll give you illustrate with one example of what we've achieved in using this genome editing technology. So this is a version of the slide that Dana showed at the very start. It shows domestication from the wolf up here on the left all the way through. And really the only important, I'm going to come this way so I can actually get the slides. The only really important bit from this is all the way through here where animals were being domesticated and in the cases of livestock, in a context for agriculture, it was completely random. There was no selection. There was no directed selection of the animals. Yes, if a disease came through or if the rains didn't happen and there was a drought or something like that, or indeed somebody who had a bigger club than you came along would affect the animals you had, but nothing. Until this man here started the policy of what's colloquially named in and in, where he just looked at the animals and said he wanted sheep with a long straight back. And he looked at the animals that had that and he put them together to mate rather than the random sort of mating that had been going on. Now, obviously, we've come beyond that selective breeding. This is the Dishley Leicester, the first animal breed that was produced through selective breeding. Now we can sequence the entire genome and use that barcode for our selection. I'll talk about that very briefly in a minute. We've been selecting animals for a long time, which I think was the inference of one of the answers to the questions a few minutes ago. We've got all these lovely animals here derived from this beast here. Some of you may feel that this is the type of pet you would want to have, but I wouldn't recommend it. Others would prefer something as stupid as this. However, they also get some useful variants of it as well. And if you look through here, you'll see this range of small dogs. One of the distinctive things is they've all got different faces, and we understand a lot now about the genetics behind them. Man has selected those by deliberately choosing the animals to breed. And we've done the same in agriculture. Here we have the sheep. Here we have a range of sheep, different types of breed of sheep. Some have got wool which are good for fine clothes. Some have got wool which are good for carpets. Others are meat, and some actually produce milk. How do we do this in agriculture? Well, we rely on genetic markers now, and initially those genetic markers were in the flags along the genome which were associated with some phenotype or other. Now we can, as I said, we can sequence the whole genome and use that as a template. We're very different from the human arena, where the outcome of the use of the technology is for the best part of the population, the overall population of animals within your herd, your flock, or whatever it might be. The actual individuals are not so important, which is obviously the reverse in the human arena. And what that means is the sequencing now, we don't sequence the full genome. We sequence maybe 1/10 of it, but we do it hundreds of thousands of times, animals, and we impute based on haplotype information and probability, the bits that are missing, and therefore we select on that big number, which is a very different approach to how you think about genetics when we talk about ourselves. So we have all this wonderful sequence information, and we have, this is called the breeder's equation. You don't need to know the details of it, but it's just that there's some really clever mathematicians who understand probability that allow us to continually improve the productivity of livestock. And that is shown in these images in the lower half of this graph, this slides here. These are all broiler chickens. We've got in essence, we've got two types of chickens, broilers, which we eat the meat off, and layers, which produce eggs. And this one here is an image of an animal on 1957. It had just under a kilogram of weight, and through using algorithms like this, markers and more now DNA sequence, we've now got just over a 4 kilogram bird to consume. And over here on this graph, shown in a different way, this is the milk production that's happened within the US dairy herd between 1960 and the turn of the century, or just past the turn of the century. You could impose, this is for dairy, you could do this for overall economic merit across agriculture. You could do it for most traits. Maybe the graph line will be slightly different and slight points of inflection across it, but that's generally what's happened. So in that context, we have, in that context, we have, that was just because I'd forgotten what my next slide was going to be. That's all. In that context, we have wonderful numerical tools, algorithms to select animals for greater productivity. I'm using the word productivity. You can apply any word or subwords to that. That could be disease resistance, it could be welfare, it could be carcass quality, overall economic merit. And we have the power and the ability to sequence those animals. And the data sets that we have with animals, especially the data sets that exist within the breeding companies, are substantially bigger than most of the medical data sets that are kicking around just now. These are vast amounts of data. We're very good at it, and this graph would indicate it's going on. The one area that we've not been very good at, even though we've pumped in tens of millions, perhaps hundreds of millions of research dollars into, or research pounds or euros, depending on which part of the world you're in, is disease resistance. The exception to that is in aqua, but that's a different story. In livestock, we are not particularly good at breeding in disease resistance. For most diseases, the clear evidence that there are animals that are less or slightly more resilient to the diseases is evident, but the idea that there are animals that are resistant to a disease is difficult to identify. And we have some really high-profile diseases to worry about. And I'm going to illustrate 3 here just to show you that although the primary impact of these diseases is on the animal, the actual overall impact is on ourselves in a variety of ways. So the first one is flu, we've heard, I'm sure you're well aware that the Spanish flu epidemic after the First World War, which spanned about 6 years, killed more people than the atrocities of the First World War did. If we had that type of epidemic today, it would probably spread around the world in about 6 months rather than the 6 years it did previously, due to the fact that we all travel. I was taken by the comment this morning that for every one diseased measles patient, they can kindly spread the virus to 12 to 18 people. I'm afraid there's enough Europeans, and I also noticed Europe was a hot spot. There's enough Europeans in this room to contaminate you, all of you. And there's only 3 of us, I think. So a big disease. We have foot and mouth disease. For any of you who were in the United Kingdom at the time we had our FMD outbreak, you'll realize that it completely changed the whole culture of people's activities. the countryside became a no-go area unless you happened to live in it. You went down the road and parked in a car park, and there was a footbath at the edge of the car park which you had to step through to go into a forest or a wood or wherever it might be, climb a hill. The impact of that is people stopped going to the country. It took about 2 years before people started going back out into the country, let alone the number of animals that had to be slaughtered. And we've heard, people are aware of post, stress from the battlefield. It's recognised that many of our soldiers and army personnel come back with that. The vets who had to kill these animals some of them are still suffering from those experiences. Then we have this one here, which is called African swine fever, and I should imagine most of you are aware of this. If I had been speaking here 5 years ago, I told you that half the pigs in the world exist in China. Well, half of those pigs are now dead, have either died from the virus or been killed. So a quarter of the pork head in the world has gone due to this one virus, which is quite staggering. We don't have the genetic understanding of the markers to go after things like that. So this is where genome editing can come in, and I explain that there. So I apologize for this to this audience, but these are slides I use when I talk to the public to try to get across genetics. I especially apologize to the geneticists in the room because you realize it's not actually true, but it's close enough. You can see where I'm going with this, can't you? So what we're talking about here is the stripe gene. And this couple have got a variant called the black gene. And dad has horizontal black and mum has vertical black. And wee Jimmy here has got a bit of both. So if that was, let's say, disease resistant for African swine fever, and these were not people, they were pigs, then this mum and dad would be spot on for what we want. We'd be able to get the offspring with the trait we wanted. However, if the trait we wanted was yellow stripe, then it would be utterly useless mum and dad. We could rely on two things. We can rely on chance to give us that mutation, and that could be today, could be tomorrow, could be in 10,000 years, or we use the wonderful tool, the tools available that genome editing technology has offered us. So we need to go after the genetic variation that can give us the traits we want in livestock. We can go into a large discussion about what those traits might be. They are just the same as the traits we're dealing with today. There's no difference except that we've deliberately engineered them. We've integrated the allele, the SNP, or whatever it might be, in one generation and only one generation. We need to go after traits which we can't easily get. I've explained to you that disease resistance is not easy to get. You heard Dana mentioned the polled locus in Holstein Friesians. You do get Holstein Friesians that do not have horns, that are polled. Except in that industry, in the dairy industry, there's a breeding pyramid where the elite animals are at the top, which all the selection goes on, and we produce our milk from the animals at the bottom. And nearly all the naturally polled animals are down the bottom. If we took them and put them into the elite nucleus, we would lose the genetic gain that's happened over the generations, perhaps 5, 10, or perhaps even 20 years of genetic gain. So we cannot do it. The variation is there, but it's impractical to use. So any variation that we cannot easily use, the genome editing tools allow us to do that. Okay, so very quickly, I'll lead into the example I'm going to do. I just want to say there's a variety of ways we can make these animals, just so that you're aware of it. They're so effective and so efficient, we can go directly into the fertilized egg or the zygote just by simply injecting into the cytoplasm. In some experiments we've done, we get 100% of our offspring edited. Alternatively, we can use cloning, somatic cell nuclear transfer. We don't have ES cells, or really robust ES cells. We're getting very, very close. We don't have them yet. And the technology that produced Dolly allows us to manipulate the cells in culture before we fuse them with the enucleated egg. We have to be slightly different in poultry. We can't get hold of the zygote. The time we see the developing embryo, there's probably 60 plus thousand cells in it. But we can get hold of PGCs, and this is work by my colleague at Roslin, Mike McGrew, where we can isolate these PGCs, we can engineer those cells, and then we can go back and produce the animals from that. And Mike's got some clever ways of making that very efficient, which I could discuss in questioning, but not as part of the talk. We can do it across all the species that we would normally recognize as farm animals, including aqua species, although they are slightly harder to do in some cases. This is another slide I use in my public engagement because there actually are 2 animals up there which have been edited. And I ask the audience to tell me which ones they are. Make a guess. Make one guess. The pigs. The fish. The pigs have. Yes, the pigs have. And this animal here has. But all those species and more have been genome edited. So the example I want to describe to you in a few minutes is work that we've been doing with a breeding company called Genus PIC. Genus teamed up with ourselves at Roslin. They also teamed up with Randy Prather's team in Missouri. And projects ran in parallel. This is the disease called porcine respiratory syndrome. It was, until ASF, the #1 disease of pigs. It still is the #1 disease of pigs because it has a wider geographical coverage than ASF has. It's estimated that over half of the farms have the disease in some form or other. In Europe, it costs us 1.5 billion euros a year. Vaccines are ineffective. We don't have any other sort of, we do vaccinate, and some places do vaccinate, but they're ineffective. We know a lot about how the virus gets into the cell and how it replicates. We don't know everything about that, but we do know some of it. And one of the players, or 2 of the players, one is CD169 and another one is CD163. I'll just focus on CD163. It is a protein that sticks through the cell wall here. This is a small internal bit, and we've got these domains that come out here, 9 domains of the protein. We know from previous cell work that this domain here is what the virus PRRS interacts with. So very simple model, and for those of you who know me well, this is not my construction. This is a much smarter postgraduate in my group. If domain 5 is there, the virus interacts with it and causes infection. If the domain's not there, the virus disappears off. So we've used 2 guides flanking exon 7, which is what encodes domain 5, to remove that domain from the animal. This is all done by zygote injection. Here we have a slide which I will amend after speaking to the last speaker. I won't tell you why, but he knows. I will amend this, and the reason why I'll amend it is because I've used the word wild type, and that's about as useful as saying it's natural or not natural. And I take the point, and I will change the slide, and one of the next ones as well. Here we have our non-edited animal. Here we have our heterozygous edited animal, and here we have a homozygous animal. And this is a Western blot showing that the protein, here's the heterozygote, here's the homozygous edited animal. And this is a histogram of the blot showing that we retain, within reason, we retain the normal levels of CD163 protein in these animals. You don't need to look at the detail here, but we've looked at the function of CD163, delta CD163 in the animals. Here it is again, I'll get rid of that. All you need to look at, I'm destroying these pointers, by the way. All you look at, they're all the same. It doesn't matter if it's homozygous or wild type or heterozygous. The graphs look the same, whether that is trafficking to the cell surface, differentiating the macrophages, or binding to hemoglobin, heptoglobin, which is the only biochemical function of the protein that we are aware of. So we've taken out domain 5, the protein is the same level, it appears to have the same function. We can produce the animals and challenge. Well, this is the outcome of the challenge study. These lines here are the non-edited and the heterozygote, and the line along the bottom is our edited animal. We see no infection whatsoever. That's the level of clinical signs. That's the level of viral RNA or antibodies produced against the viral, nothing. And the team in Missouri see exactly the same thing. They've taken a slightly different approach. They've created a null, they believe they've created a null allele by destroying the function of the gene. Proving you've got a true null is very difficult to find the protein that may or may not be there, but they've not found any protein using available antibodies. So for the first time, we've gone in and created virus-resistant pigs. Remember I said at the beginning, there's hundreds of thousands of pounds being put into looking at virus resistance. Well, that's exactly the case for PRRS, and there still are ongoing large projects here in the US and in Europe looking at PRRS resistance. None of that work, none of that really good genetics work, it's very, very good work, none of that is thrown up that CD163 might be a candidate. This is purely done by virology, empirical in cells, finding a cell which is either tolerant to the virus, allows the virus in, or doesn't. So in the absence of biology of the cell, in fact, these targets come up. And this is the approach we're going, a part of the approach we're going chasing after other targets. So for those of you like myself had some bacon for breakfast, maybe in a few years' time, this will be where the bacon comes from. I'll finish there by saying that there are a number of people involved in the project. I've just named a few there. We've obviously got the farm hands and obviously the animals themselves. Thank you for your attention.